quantify sleep apnea in mouse models of Down syndrome. You see, it just rotates here.
Okay, so it does what?
What it does, it just rotates, and then this has some kind of infrared sensor. It quantifies the blood oxygenation and heart rate and respiratory rate, all this at the same time. And then these sensors quantify the movement of the mouse. That's why they count how much the mouse moves. Because everybody with Down syndrome, almost 70 to 100% of people with Down syndrome suffer from sleep apnea.
Oh, really?
Yes. And the question is that, but we are trying to see whether if you try to improve sleep apnea in these individuals, whether that would improve their cognition, and that's the idea.
Okay, I remember when you gave the presentation, you said something about short-term memory processing into long-term memory during the sleep stage.
Right.
Could you talk a little bit more about that?
Right. I mean, we say that the area that they modulate this newly acquired information is the hippocampus, and that area undergoes significant degeneration in people with Down syndrome. And then what we are trying to see whether we can restore the function of that region.
Okay, can you tell me about the correlation of sleep apnea to that?
Well, I mean, the idea is that, as I say, 70 to 100% of people with Down syndrome have a sleep apnea. And then when you have a sleep apnea, the amount of oxygen that goes from the lungs to the brain significantly goes down. And then that causes significant failure and cellular injury, which eventually we hypothesize that it causes, cognitive dysfunction and loss of memory in people with Down syndrome. But we are saying that chronically for a very long time, when the amount of oxygen going to the brain is a little bit less, that would eventually lead to further and further deterioration of cognition in people with Down syndrome. And the idea is that, is there any way to fix it? And then is there any way to identify the gene that's causing that? That's the whole idea.
Very interesting.
But you see that this is a machine that just built to have the balance, and once we put the mouse into here, and then the mouse can easily move around, and then we live record the heart rate, respiratory rate, the blood oxygenation, all the movements, everything we record at night while we are not there. While we are not there, the mouse sitting there and then is not stressed at all. And then we quantify whether when the mouse is supposed to sleep versus when the mouse is not supposed to sleep, whether there's a difference in terms of this type of sleep apnea that they have.
And how much they retain or anything?
How much they retain, and whether there's a loss in the amount of oxygen that gets to the brain.
Do you have them perform specific tasks or anything like that?
Right now, not here. Not at all. We are just trying to not stress them. I mean, that's the biggest thing. That's why we just let it go, and we program a computer to start recording while we are gone for a kind of while. They feel confident that there's no danger or nothing, and the mouse can go, has access to water and access to food, and then we start recording, and there's absolutely no distress for the mouse, and that's
so kind of like mapping the biochemical process in a natural state.
Right. I mean, we're just trying to temporary, meaning that we just see if there is a failure in cognition or if there's a failure in respiratory rate. When does it happen? Does it happen during the night? Does it happen during the day? Does it happen when they're supposed to be asleep? Does it happen when they're supposed to be awake? Or does it happen when they run a lot? Or does it happen when they're resting? And that's what we're trying to understand.
Interesting. What else do you have for us?
Oh, see, we have a very sophisticated system in terms of reconstructing the regions that undergo degeneration in people with, in mouse models of Down syndrome. We have to cut these sections one by one and stain them, and then this machine allows us to reconstruct it back and rebuild it entirely back in 3D, and then analyze it all at the same time. That's the whole idea.
What have you learned?
Oh, what we have learned is that these neurons communicate with each other through dendrites, and then we realize that there is a significant shortening of these dendrites in mouse models of Down syndrome. If you look here, you see, this is it.
Where am I looking?
Yeah, this is it. These are the dendritic tree arborization in mouse models of. This is for the mouse model of Down syndrome, and this is a mouse model. This is a normal mouse. And you see, these dendrites are the branching that is supposed to communicate with each other and transfer information from one side to another side. And as you see, these are much shorter and much less complex compared with this one. This is much bigger, much longer, and they're much complex. That's why they make more communication. And when there's more communication, they can make more synapses, and once you have more synapses, you can do a better cognition and a memory, it's significant enhancement in memory. That's the whole idea. We realized that there's a significant reduction of these in, in mouse models of Down syndrome. And this one actually shows these cells, these are the cells that we reconstructed here. We follow these cells, and these are the cells that we just go, these are dendrites, and then follow them 3D and reconstruct them and then put them together, and then we build that.
Interesting. Yeah, in case the audience doesn't know, they can induce Down syndrome into mice.
Yes, we can. Yeah, we can. Because most genes that are on human chromosome 21, they can be found on mouse chromosome 16. That's why we have a mouse that has most of the genes that are triplicated in Down syndrome, also triplicated in these mice. And then these mice, in many aspects, quite similar to Down syndrome. They have memory problems. They have some indication of sleep apnea. They have a lot of problems in terms of degeneration and atrophy and shrinkage of the cells in the regions that are major players in cognition. That's why in many aspects these mice are very useful for us to do that. Why? Because if we identify a drug, first we have to go and try it in these mice and say, does it work? And what would be the best dose? What would be the best regimen? And then once we learn that, okay, we have a good, effective, safe drug, then we take that and use that in, eventually we start a clinical trial.
Mm-hmm. And this research is not specifically handcuffed to Down syndrome, but it's more along the lines of towards the brain, how the brain works, and different types of defects with the brain or conditions of the brain, which can be applied to other types of conditions as well, right?
Oh, absolutely. The best example is Alzheimer's disease, because almost every individual with Down syndrome at the age of 40 will show a brain that is absolutely indistinguishable from a brain of a person with Alzheimer's disease. And that's why, and then most of these individuals eventually will get dementia of Alzheimer's type. That's why this indicates that there's a very good similarities between Down syndrome and Alzheimer's disease. If you learn how Down syndrome happens and what are the genes that are major players, and if you can bring down their expression, you have a very good clue to understand what happens in Alzheimer's disease.
Mm-hmm. So to be clear, you're not necessarily saying we want to cure this disease or this condition that we think is, that people don't want, but you're just trying to improve quality of life and allow individuals to live a better life with the life that they have.
Right. Because the only thing that we target is learning and memory, and that's what we want to do. We want to see whether is there any way to improve learning and memory in mice first, and then in humans with Down syndrome. That's it. That's all we want to do. We want to make them do better at school. We want to learn better new tasks, and be able to live independently and have normal intellectual activities.
I like that.
Great.
Anything else we have?
No, great. Thank you.